Okay. So now for the gene control, we're going to look at this. Okay, we're going to distinguish between structural genes and regulatory genes. Okay, and between the repressible and inducible enzyme. Now, this is quite a new topic for you guys. A lot of terms that you may not be heard it before. Doesn't matter. Later, I will show you one by one. Okay. Then explain the genetic control of the protein production in prokaryotic cell using operons called Lac Operons. The Lacs here means lactose. Okay. Okay. It's lactose operons. Okay. And then so we're going to look at in the eukaryotic cells, we have a special protein here called transcription factor. Okay? We call it transcription factor. We're going to look at this. What is the function of transcription factor and why we need to have a transcription factor? And we're going to use these as a sample, DELA protein as a sample, as a repressor, and we're going to look at how this can have the effects onto the transcriptions by one of the plant's hormones called gibberellin. Okay, so this part totally new for you guys, so please do follow. Okay, now we look at gene control in the prokaryotic cells. Okay, if you put it this way, very simple to study prokaryotic cells because they are unicellular. So easier for us to study them because there's no cell-cell interactions. Okay, and means that when they have the genes, they will be able to give us a protein. Okay, but the question here is they don't simply give us a protein, but the response to the environmental change. So this environmental change, we always call it or known as stimulus or stimuli. Okay, so how it respond? Respond by switch on or switch off a particular gene. Okay, switch on or switch off a particular gene in the metabolic reaction. When they need a particular enzyme, they switch the particular gene on. Okay, so for example, if let's say in the presence of the lactose, so it means that you want to convert lactose into a glucose plus galactose, you need the enzyme lactase, for example. Are you clear? Okay, or beta galactase, galactosidase, for bacteria. Okay, this in humans lah. Okay, in bacteria we beta galactosidase, the enzyme involved. So means that if there is no lactose, then the bacteria won't produce beta galactosidase, but the gene is still there. So how they respond to the lactose? Lactose present means that there is a change in the environmental factor, right? So when the lactose present means that there is a change in the environmental factor. Lactose is an inducer here. Lactose is a stimulus. Stimulate what? Stimulates the bacteria to produce the enzyme beta galactosidase, therefore can hydrolyze. If there is no lactose, then the gene won't be switched on. So today we're going to learn how this gene can be switched on and switched off in prokaryotic cells. Okay. So cell can adjust again the production level of certain enzyme by regulating the expressions of the gene and code for the enzyme. So the control enzyme production can be either at the level of transcriptions, means the synthesis of mRNA, which code for these enzymes. Okay, so if always remember when say expression take place, when particular gene expression take place, the first thing we must have mRNA. If no mRNA, means that's no gene expressions. Okay, so we're going to learn these two in our syllabus, only Lac Operon, which is inducible operon. Okay, Tryptophan Operon is not in our syllabus, not in syllabus. But we still will talk about it. Why we want to still talk about it? Why? Because in the learning outcome here, we need to know what is mean repressible enzyme. Can I see that? Inducible, no problem. We're going to study it. Repressible enzyme, we're going to use this tryptophan operons to learn about this. Okay. So now we start with the first one, the Lac Operons, which is inducible operons. So in this operon, what's mean operons? Now, if you look at this structure, let's go to this. I'm going to draw it out. For bacterial, okay, you have the gene. Okay, bacteria have the gene. So this gene, we call them as a structural gene. So these structural genes can be transcribed to form mRNA, and you give us a protein, a polypeptide first. So this polypeptide will fold into either become enzyme, or fold becomes a hormone, okay, or fold becomes the protein, okay. So involved in the functions of the cell, or we call metabolic functions in the cells. Can I see that? So this gene, we call them a structural gene. Okay, understand, huh? Structural gene. So sometimes you may have a cluster of the gene. For example, in the Lac Operon, you have 3 genes together. So it means once we switch on 3 genes together. So they are in a cluster. Okay, so this is the structural gene. Now, for a gene to function, to be transcribed, then they need what we call as the promoter. So what is the function of promoter? Okay, promoter actually is very, very important. Always remember the function of promoter here. Okay, for any gene to be transcribed, must have promoter. So all genes must have promoter. So what is the function of promoter? Promoter actually, if you still remember in the protein synthesis, we always learn about RNA polymerase. We learn about RNA polymerase. RNA polymerase is the one that initiate what? Transcription. Okay, initiate transcription. Okay, but how the RNA polymerase know which gene to initiate, which gene don't? So it depends on the promoter. So promoter actually is the binding site of what? Binding sites of the RNA polymerase, so that it can initiate, okay, synthesis of mRNA or transcription. So it means that the gene that without promoter actually useless. The genes that without promoter actually useless. So between the gene, structural gene, and promoter, we have a special sequence here called operator. Now, operator is optional. Operator is optional, not necessary, but promoter is the must. So what's operator? Operator actually act as a switch. Okay, act as a switch here. So in general analogy, okay, so if you have a lamp or light source, okay, as long as you connect to the battery, it will be turned on. It will continue to turn on. So analogy here means that this is the gene. So the gene always switch on. But the problem here is, who is the promoter? Battery is a promoter. So it means that if you have the light, the gene, then without promoter, basically you have the light bulb without the battery. So it won't switch on in this case. Are you clear? But in order to switch it on or switch it off, if you want to control, gene control in this case, you want to have a control, so you need to add a switch. So this switch actually is the operator. Can I see that? Then I can control when I want to turn it on. For example, if let's say the room is very bright, so then I switch it off. Right? If the room is very dark, then I switch it on. So it means the operator will help us to control whether the gene have to be switched on or switch off. So use this as an analogy, then you understand the concept here for all the genes, they must have the promoter. Why do you need to have the promoter? Because promoter is the binding site of the RNA polymerase, allowing the gene to be transcribed. Okay, if without operator, can or not? Yes, can. Without operator basically means that without the switch. Without the switch, yes, the gene always switch on. So when it always switch on, means that whether I need it or do not need it, it's going to continue to switch on. So it's full in this case, right? So that's why some of the gene, not all, some of the gene, they have the operator. So this operator will act as a switch. So later we're going to look at this, how they functions together. Okay. So now look at this. So the whole thing here, from the promoter all the way until the structural genes, this one we call it as the operon. So operon is basically a functional unit for the genes so that they can actually code for a particular polypeptide which can fold into the enzyme, hormones, protein that involve in a certain functions. Okay. Now, most of the times, okay, in order to have these effects onto operator, in front of the structural genes, you can see that we have a regulatory sequence. So means that dotted means that don't care what is the sequence, but you can see that you have this gene we call as a regulatory gene. Okay, regulatory genes. So every genes, they must have their own promoter. So it means that they have their own promoter. So this promoter, can you see that? Without operator. So this regulatory gene always switch on. Okay, so you compare just now one of the parts we need to compare, structural gene. Structural gene actually code for the polypeptide. So this polypeptide can form enzyme, hormones, protein, which involve in the cellular functions or metabolic functions or cellular functions. Okay, but how about this regulatory gene? Let me remove this one first. So what is the function of regulatory gene? So as long as they are the gene, they code for protein. So they get mRNA. This mRNA will going to be translated to form a polypeptide. So this polypeptide going to be to form what we call the repressor protein. When this repressor protein is active form, what we're going to do, this repressor going to bind to the operator. Okay, try to imagine, they bind to operator. So sometimes these repressor protein are quite large. When they bind to operator, can you see that they block the promoter? You realize that they block the promoter, so means that RNA polymerase cannot bind to the promoter anymore. Realize that not? So what's the function of regulatory gene? Regulatory genes always produce the repressor protein because they have the promoter. RNA polymerase can bind. When the RNA polymerase can bind, so this regulatory gene always express. When express, you get the mRNA, you get the polypeptides, and then from this polypeptide, we're going to get the repressor protein. Active form repressor protein going to bind to the operator. Therefore, RNA polymerase cannot bind to the promoter, so no transcription. Are you clear? No transcription. But certain stimulus can help us to remove. Later we're going to learn. When you remove this repressor protein, then RNA polymerase can bind. When RNA polymerase can bind, then we have the transcription. We're going to have the polypeptides, and then cellular functions actually take place. So this is a general idea about how the operon actually functions. Okay, how the operon actually functions. Okay, so what you need to do here, what you need to know. So first thing, you need to differentiate between structural genes and regulatory genes. So regulatory genes, the protein is a repressor protein. They do not involve in the cellular functions. No, they don't involve cellular functions at all. So what they do, they actually regulate, right? To regulate the expressions of the structural gene. Structural genes, they code for the mRNA, which can be translated to a polypeptide, which code for the enzyme, hormones, or fold into proteins. They give us cellular functions. Okay. So come back to our notes here, then you understand better. So the components of the Lac Operons, you need to know, we have the promoter, followed by operator, and the structural gene. Okay, so if you look at these diagrams, you can see that here, the first part, the red color is the promoter, brown color operator, and then we have 3 genes here clustered together called Lac Z, Lac Y, and Lac A, 3 genes. So it means that these 3 genes will be expressed together. Okay, so the entire thing here is known as the Lac Operons. It's a Lac Operons. Can I see that? Okay, so to control the Lac Operon, we need to have the repressor genes or regulatory genes. Okay, in our syllabus, we call it regulatory gene. So these regulatory genes can see that transcription always takes place to get the repressor protein. So this repressor protein bind to the operator. Therefore, these operons actually now switch off. Why switch off? Because RNA polymerase cannot bind. RNA polymerase cannot bind. Therefore, no transcriptions. Okay. So what's the function of promoter? Promoter highlight is allow the binding of the RNA polymerase so that transcription can start. Okay. So what's operator? Operator is a DNA segment allow the binding of the active repressor protein. So once it bound, it blocks the RNA polymerase. So it's something as a switch. Can you see that? When it block the RNA polymerase to the promoter, therefore the gene expression off. Okay, gene expressions off. Okay, so structural genes. Structural genes here, for Lac Operons, it code for the polypeptide. As long as the gene, they code for polypeptide required by the cells. So considers 3, Lac Z, Lac Y, and Lac A. So you need to remember the name because past paper come out before. Okay, Lac Z, this is my mistake, it's a beta galactosidase, not 3. It's a beta galactosidase. Lac Y code for the permease, and Lac A code for trans-acetylase. Okay, so the 3 gene. So what is the function of regulatory genes? Regulatory genes. Regulatory genes actually code for the polypeptides that regulates expressions of other genes. So that's why we call regulatory genes. They code for what? They code for the repressor protein. So when they're active, they're going to bind to the operator. Okay. So what's so special about this repressor protein? Now look at this. Look at this diagram. Again, look at this diagram. Now, when we do have the operator bound, okay, can you see that? Operator bind with the active repressor. Therefore, RNA polymerase cannot bind, or it can bind, but it cannot initiate the transcription. So this operator will have a special site here called allosteric site or binding site. So what's so special about these allosteric sites? Look at this diagram now. Look at this diagram. So how we switch it on? So now look at this. No lactose. So it means that we don't need enzyme. When no need enzyme, so it means that we switch off. Okay, we switch off the operon. But now with the presence of lactose, we need enzyme. So we need enzyme means that we need to switch on. So how we switch on? Very simple. Now look at this. We have the lactose. Okay, allolactose or lactose. So when this lactose, can you see that? When this lactose bind to the repressor protein. So it changed the shape of the repressor protein. So this shape now can see that they no longer can bind to the operator. Right? Cannot bind the operator already. When it cannot bind the operator, RNA polymerase now can bind to the, can I see that? It can bind to the promoter. Therefore, transcription take place, allow us to have three enzymes here: beta-galactosidase, permease, and transacetylase. So beta-galactosidase hydrolyze lactose. Permease increase the permeability. Transacetylase, it transfer the acetyl groups. Okay, these transfer acetyl groups. So in our syllabus, we need to know these two. Increase the permeability, allow the lactose to be absorbed into the cells. Can you see that? We switch on already. Do you realize that? Because of the presence of the lactose, let's say we put the bacteria in the medium that contain lactose. When they have lactose, what will they do? This lactose will bind to the repressor protein. When they bind to repressor protein, now the repressor proteins become inactive. When they're inactive, they cannot bind to the operator. When they cannot bind to the operator, RNA polymerase bind to the promoter, initiate the transcription, allow us to have three enzymes which involve in the lactose metabolism. Okay, breakdown of the lactose. Okay, can I see that? Now, but when you have the beta-galactosidase to hydrolyze the lactose, then the concentration of lactose decreasing. When the concentration decreasing already, so means that lack of the lactose to bind to repressor protein. So repressor protein now have chance to bind to the operator and then switch off the gene expressions. Can you see that? It's a negative feedback mechanism. Eh? It's a negative feedback mechanism. So how? So when lactose is high, we need enzyme. So we need to switch on the Lac Operons. So how we switch on the Lac Operon? So it means that lactose, because present of lactose in high concentration, higher chance for lactose to bind to the allosteric sites of the repressor protein, turn the repressor protein become inactive. So this inactive repressor protein will detach or it cannot bind to the operator. RNA polymerase now can bind to the promoter and start the transcription process, allow us to have the enzyme that involve in the metabolism of the lactose. Okay, so when we do have the enzyme to hydrolyze the lactose, the concentration of lactose decrease. So when the concentration of lactose decreases, so lactose have less ability or less chance to bind to the repressor protein. So it means that some of these repressor protein have chance to bind to the operator, stop the gene expression. So this is a form of negative feedback mechanism, eh? A form of negative feedback mechanism. Okay. So look at the, you see. So therefore, this regulatory genes and repressor protein act as the negative control system. So if there's no lactose, regular gene function to produce a repressor, bind to the operator, stop the synthesis of the enzyme. So if lactose present, repressor protein detach from the operator, start the production of the necessary enzyme. So absence of lactose will result in no enzyme. Therefore, the bacteria actually save their ATP, right? Because we don't need. Like for example, the room is bright already. Why you want to turn on the light, right? Okay, so same concept here, right? Same concept. So you can arrange the events. Okay, so I'll show you the answer. So understand this, okay? So therefore, if you look at the Lac Operon, Lac Operons code for the enzyme. So this enzyme, we induce it, right? So this enzyme, we call them as inducible enzyme. So what's mean inducible enzyme? So when talk inducible enzyme, under normal conditions, without lactose, you won't have the enzyme. Or we say that only present when inducer present. When you have the inducer, in this case, lactose. When the lactose present, yes, you induce the enzyme. So that's now enzyme is made. Okay, so this is the idea of what we call the inducible enzyme. Now, tryptophans. Okay, tryptophans it's not in our syllabus. It's for informations only for the entire thing. but we need to know what is meant by the repressible. Repressible enzyme basically means that the presence of the tryptophan stop the operon. Basically means that under normal conditions, they are switched on. Are you clear? So with, that means without tryptophan. But with tryptophan, then we have to switch off. Okay. So we're going to look at the principle why. Okay, why in this case. So if you understand the concept of the lactose operons or Lac Operon, tryptophan operons shouldn't be a problem for you guys. Okay. So what's tryptophan operon? Tryptophan operon is involved in the synthesis of the amino acid called tryptophan. Same as the lactose operon or Lac Operons, they have promoter, operator. Instead of 3 structural genes, they have 5 structural genes. Even though 5 structural genes, but they code for 3 enzymes. They code for 3 enzymes. So enzyme A, B combined together form 1 enzyme. C itself form 1 enzyme. E and D combined together form enzyme. So total we have 3 enzymes. So these 3 enzymes are involved in the formations or synthesis of tryptophan, the amino acid tryptophan. So means that under normal condition, when there's no tryptophan, so this operon going to be switched on. Are you clear? Why? Because not enough tryptophan, right? Bacteria produce their own tryptophan, switch on. So how we going to switch on? If you look at the tryptophan operons here, you can see that this is a structural gene, this is operator, this is a promoter. Under normal conditions, the repressor protein is an inactive repressor. So it means that they produce, can you see that? The regulatory genes produce inactive repressor. So this inactive repressor cannot bind to the operator. It cannot bind to operator. So when it cannot bind to operator, yeah, good. RNA polymerase is going to bind to a promoter and continue. Okay. So this diagram a little bit wrong. Promoters is only here. Okay. Not this part. Okay. Promoter. This one is operator. Okay. So this is extra information for you guys. How we inhibit? We can inhibit in the we can inhibit at the protein level and inhibit at the gene level. So how we inhibit the gene level? Okay, let us look at the regulatory gene. So regulatory genes here is away from tryptophan operons, not totally nearby. So again, they code for what? They code for repressor protein. So what's this repressor protein? Repressor protein actually inactive. So repressor also have allosteric proteins sites to be bound by the tryptophan. So once the tryptophan bind, it become activated. Okay, so it depends what? It depends on the nutrients. Okay, so what does this mean? Now look at this. Let me do this quick one. So this is structural gene for tryptophan operon. SG stand for structural gene. And then they have operator. They have the promoter. So distant away from this tryptophan operon, you have the regulatory genes and also the promoter. So regulatory genes here, always you get the mRNA, you always get the repressor protein. Okay, but this repressor protein is inactive. So they cannot bind, okay, to the operator. So this inactive, they cannot bind to operator. Okay, they cannot bind the operator. So therefore, RNA polymerase can bind. And then transcription take place, allow us to get the mRNA of the structural genes, and then we get the polypeptide, and we'll be able to get the enzyme. So this enzyme for what? This enzyme actually allowed the bacteria to get or to form tryptophan. Tryptophan TRP. Okay, tryptophan. Okay, so now why? In this case, because bacterial needs tryptophan. Okay, need tryptophan. But now, if let's say we put this bacteria in a surrounding that with tryptophan, what happened here is tryptophan will bind to the repressor protein. Okay, tryptophan will bind to the repressor protein, from inactive stage become active. When it become active already, then this repressor protein will bind to the operator, stop the binding of the RNA polymerase. So therefore, you're going to stop the productions of the tryptophan, which is true because when you put the bacteria in the medium that contain tryptophan, they do not need to produce tryptophan. I mean, the enzyme that help to synthesize the tryptophan. Correct? Logical thinking. Now, in this case, if you put the bacteria in the medium or the environment full with the tryptophan, do they need to produce tryptophan? They do not need to produce tryptophan. So they have to switch off. So how is switch off? Because of tryptophan, high concentration of tryptophan. So with a high concentration of tryptophan bind to the repressor protein, make it become active. Okay, make it become active. So when make it become active already, so this active repressor protein will bind to the operator. When they bind to the operator, RNA polymerase cannot bind to the promoter, therefore no transcription process takes place. They do not need to produce enzyme to make tryptophan. Why? Because they already with high tryptophan. Okay, but the cell will use tryptophan. Right, the cells will use tryptophan. So the concentration of tryptophans decrease. So when the concentration of tryptophan decrease already, then less chance for tryptophan to bind to repressor protein. Repressor protein become inactive. Therefore, this operons is switched on again. Can you see that? So therefore, the enzyme involved here we call as a repressible enzyme. Because under normal conditions, they are there. But we can actually stop the productions of the enzyme, right? With the presence of the inducer here called tryptophan or co-repressor here. Are you clear? So this tryptophan we call as a co-repressor. So with the presence of co-repressor here, we will be able to stop the productions of the tryptophan. Okay. Okay. So look at this tryptophan operon. So just now we say that, okay, these repressor proteins, they are inactive, okay, inactive. So therefore, they cannot bind to the operator. So RNA polymerase will bind to the promoter and transcription process take place. We get the enzyme to form tryptophan, right? But now, with the presence of the tryptophan, which act as a co-repressor, when they bind, it turns this repressor protein become the active form. Why? Because when they bind to operator, the active repressor protein bind to the operator, therefore RNA polymerase cannot bind to the promoter, therefore no transcriptions take place. Why with no transcription? Logical thinking, because we already have the tryptophan. So when we already have the tryptophan, we do not need to form more tryptophan. Until the cell use more tryptophan already, then no more tryptophan to bind to the inactive repressor. Okay, therefore this inactive repressor now cannot be active, or this active repressor now cannot bind to the operator. Therefore, transcription process can take place. Okay. So I show you the answer here. So unbind itself is always switched on. RNA polymerase can now bind to a promoter. It start the transcription process to take place. Okay. So unless we do have this regulatory, genes, so the tryptophan repressor is synthesized in active form. So when they bind, then it can change the shape by this co-repressor. Therefore, it can bind to the operator. Okay. So we call it a repressible operon because transcription usually on, but can be inhibited. Okay. So we have done for today. Okay, I'm not going into this gene control in the eukaryotes for today. We're going to look at it in next sessions. Okay, so a quick summary here. Three things we're going to talk about here. Three things. So we look at the Lac Operon compared to the Tryptophan Operon. First thing, we look at the repressor protein. So repressor protein, okay, original form. This original form of repressor protein for the Lac Operons, they are active. Okay, but Tryptophan Operons is inactive. So active means that they bind to operator. So switch off. Inactive, so we won't bind to operator. So this one switch on. Okay. So the effect of inducer or co-repressor on repressor protein. So what will happen here? If what's the effect? So this one going to inactivate repressor protein. Are you clear? Okay. So when it inactivate the repressor protein, so you switch on. So okay, so you switch on the operons. But in this case, it activates the repressor protein. So when activate repressor proteins, then it switch off. And I see that, okay, the effect. So what is the type of the operon? This one is inducible. Why inducible? Because under normal conditions, it's off. So only with the presence of the lactose on. The idea here, why? Because we want to hydrolyze the lactose, we need the enzyme. This one is the repressible. Okay, so it means that under normal conditions, without repressor protein, they are on. But with the repressor, with the co-repressor, okay, so with the tryptophan, so it switch off. So lactose act as the inducer here. Tryptophan act as a co-repressor. Okay, so hope you guys understand what is the meaning of inducible enzyme and the repressible enzyme. If you want, you can take a photo. Okay, and that's all for today. Okay.